So, hi everyone. Hi everyone. So I'm going to talk about my project. I changed the title a little bit just because I've been focusing more on all African great apes so far, and on the second part, during the second part of my PhD, I will focus more on mountain gorillas. So. Okay, so as I just said, my target species are African grey apes belonging to the two genera Pan, which includes bonobo and chimpanzees, and gorilla. There are 4 different species, bonobo, chimpanzees, and gorillas, and 8 different subspecies occurring in Western and Central Africa in forest, mountain, and savanna ecosystems, where they play a fundamental role for the maintenance of the ecosystem, and they perform vital functions such as seed dispersal, which is crucial for the recruitment of trees, and also for the maintenance of the heterogeneity of the habitat, so that other animal communities can thrive in the same environment. Despite the ecological importance, they're still listed as either endangered or critically endangered by the International Union for Conservation of Nature, with the main reason being the continuous decline in the number of population numbers throughout the home range. The major threats are poaching, diseases like the Ebola virus, habitat loss and degradation, human-wildlife conflicts, and low genetic diversity, which diminishes their ability to face and withstand stochastic events like pandemics and climate change. But because of these, it is extremely important from a conservation perspective to monitor changes in populations. And there are different methods that can be used. For example, there's distance sampling, like nest counting, or camera trapping. There are also other methods. So these methods have been applied for years now, and they're fairly simple to use, but they all involve some sort of uncertainty when it comes to individual identification, and so to accurate counting. And these can be a problem when your main aim is to assess changes in population numbers. There's also of course, as I just said, there are other methods that I'm not describing here, but can be found on dedicated websites. But there is another method which is still under development, and that is genetic survey. Genetic survey is done via DNA testing. Compared to other methods, it's efficient, and indeed the cost of running DNA testing is decreasing year after year, and it's more precise. Once you've managed to profile, which means identify all your individuals, then you can, of course, access accurate counting, but also start to analyze population dynamics, such as gene flow, and understand how individuals move from one group to the other within the home range. And there's also another important aspect that is not discussed very often, which is less disturbance to the animals compared to other methods. And indeed, new technologies allow you to exploit non-invasive samples, such as hair and fecal samples. So on the right here, I'm showing the phylogeny of African grey apes, including humans. So it is important to keep in mind that African grey apes are our closest relatives, sharing more than 98% of the genome with us. Here, below in the slide, is a simplified visual representation of DNA testing for individual profiling. So you get a DNA, you sequence it, and you got the profiles of your individuals. So having said so, the aim of my project is to exploit the latest DNA sequencing technology so part 1 is to test the potential for grey ape species and individual identification of a commonly used human-based forensic kit developed by Illumina. So basically what this means is that for the first time ever, we are testing a forensic kit designed for human investigation in known human grey ape species for conservation purposes. And this is to help conservationists to exploit a tool that is already there and has already been developed, to exploit this tool to conduct genetic surveys for conservation purposes. The test was run on a sample set of 54 different individuals belonging to 5 different groups of African grey apes. And then the second part of my PhD, which we start soon enough, and is perhaps the most exciting bit, at least in my opinion, is the development of a new kit that would allow me to work directly in the field and run the DNA testing directly in the field, so without the need of any particular laboratory facility. And I'm going to explain this a bit more in detail later on. So as I said, I've been using a human kit which contains markers, and in this case, these markers, which are known regions of the genomes that are used for DNA testing, these markers are shorthanded repeats, or SCRs, which are multi-allelic, fast-mutating repetitive region. Poorly speaking, SCRs are regions, known regions of DNA made of blocks repeated in tandem. So you can see here that SCR1 for individual 1 is made of block AGAT repeated 4 times, whereas the same repeat for individual 2 is made of blocks repeated 9 times. By looking at several different SCRs at the same time, then you are able to distinguish different individuals. This is because they work very much like fingerprints. So again, the pros of using SCRs as markers for DNA testing is that they're very good for individual identification. And also, and this is really helpful for me, is that they're well studied in humans. So when it comes for me to make comparison between grey apes and humans, I have data that I can exploit, and that is already there, and I can exploit for my study and research. The cons of using these markers is that because they're so fast mutating, they change quite rapidly. And so it's easy enough for you to tell different individuals apart, but it's really hard for you to assign individuals to the correct species. And this is mainly due to the fact that they're normally typed by a capillary electrophoresis, or CE, which gives you information on the length of your SCR. So in this case, it would tell you what your SCRs is 4 repeat long, and in individual 1, and it's 9 repeats long in individual 2. And also there's a chance that these repeats may be quite long, and so you need very good quality DNA, which is not always the case when you're dealing with non-invasive samples. It can be fragmented, so chances are that your SCRs are fragmented in the middle. So an example of the issue I've been discussing is here. So here, for example, I've been analyzing 3 different individuals belonging to 3 different species of gorillas. And if I use the length-based information only that derived from capillary electrophoresis studies, I can see there's no difference across my samples. So it's impossible for me to distinguish these samples, let alone assign them to the correct species. With the development of massively parallel sequencing, however, or MPS, I can now access STR structure information, and I can perform a more detailed analysis. So this is the kind of information I would get from MPS. So in this case, you see each color represents a different block. So say, for example, purple is a block made of the letters AGAT, orange is ACCD, yellow is a different one, and so on and so forth. So you can see here that using information deriving from MPS, I can easily tell individuals apart and assign them to the correct species. So the results of that we have obtained so far are really promising. So first of all, the kit worked, and indeed 25 out of the 27 STRs contained in the kit amplified, meaning they worked correctly across species, 13 of which worked in all individuals and were good enough to allow me to distinguish different individuals. So the important findings here is that not only did the kit work, but it also allowed us to distinguish different individuals. And even more exciting is that we were also able to assign individuals to particular species of belonging via clustering analysis. And we're now starting also some sort of phylogenetic analysis, but this is still at a very basic and initial stage. So here's the result of the clustering analysis done via DAPC, which is an improved version of PCA analysis that maximizes differences across groups. We can see, so each dot here represents a single individual, and the ellipsis is the clusters of belonging. So we can see that all individuals were assigned to the correct species because they all fall within very close to the ellipsis. And also gorillas were clustered close together. So yeah, we were able to assign the species. And this is a major breakthrough because you have to keep in mind, this is the first time that anything like this was ever done. So we're really happy and excited about this finding. Okay, so for the second part of my PhD, as I said, this will involve the development of a novel kit containing a different set of markers. So we won't be using STRs, but we'll be using SNPs, or single nucleotide polymorphisms. These are basically, poorly speaking, single base change. So as you can see here, individual 1 and individual 2 are basically identical, apart from the base per AT in individual 1, which changed to GC in individual 2. So these kind of mutations, they don't happen as fast as SCRs, so you need more SNPs to have the same discrimination power of a single SCRs. But the good thing is that because they don't mutate so fast, it's very good for species identification. So by increasing the number of markers, the number of SNPs we look at every single time, we can get information both on the individual and on the species on belonging. And the biggest advantage of all is that in this case, your DNA doesn't need to be a very good quality because even if it is highly fragmented, like the DNA that of course in fecal samples, for example, is highly fragmented. It doesn't really matter because it's still only a single base change. So even if your DNA is reduced to very short fragments, this method will still work. And the other advantage is that this kind of kit would be could be used in portable devices like the Oxford Nanopore mean iron, which is this device that you see here is basically a portable device that allows you to do DNA testing directly in the field. So you don't need any kind of, in theory, you don't need any kind of laboratory facility. So I can easily travel to Uganda, Congo, wherever, and perform my analysis directly in the field. This is also easier from a methodological point of view. So even known trained people can then run DNA testing. So this is really breakthrough for conservation in underdeveloped countries where no good laboratories are present. So yeah, so this is what we are trying to do, and these are our major results so far. Thank you very much for listening. Any questions?